A couple of things. I know I mentioned this in passing, but just a heads up, no homework over the weekend. So if anything, study immunology, because I don't want to get to next week and then have everybody flip out on Wednesday and go, oh shit, we have a final coming up in 4 days, and oh shit, I forgot everything about immunology. So it makes a big appearance. So let's not have a repeat of last exam, I suppose. No, hey.
Is that a threat?
It was just a comment. It wasn't at anybody in particular. It could have been at myself.
The whole class.
All right. Other than that, plan Wednesday next week. Review 4 to 6. Okay. I'm going to be around on Monday. I'll be around Tuesday. I'll be around Wednesday morning as well. So you're always welcome. And sorry, I should rephrase that. During finals week, I will be around all day Monday, Tuesday, Wednesday. But of course, let me know if you've got questions. I will tape the review as always. And I'll meet with anybody outside of that time too if you want to. All right. I think that's about all I wanted to cover. The vaccination slides and mini lectures and stuff, I will be working on getting those up over the weekend so that by end of the weekend, Monday morning, they're up and ready to go for you. And then it will be our last section. So what I wanted to do today, I said I wasn't going to do a case study, or originally I was going to do a case study, and then I said, nah, I won't do one. And then last night it became kind of like is a partial case study. So what we're going to do today is we're actually going to start by talking about Streptococcus pneumoniae and talking about how does it actually cause some of the signs and symptoms of meningitis. So particularly what we're going to focus today's attention on are the virulence factors, or the proteins and molecules that this organism produces that is required for it to replicate inside of a host. Okay? And then we'll talk about some of the effects of these. But the way that I want to start this out is just to kind of walk through in about 15 to 20 minutes, just walk through how do we go from infection to the point where strep pneumoniae breaks the blood-brain barrier. And that's where it transitions from an organism that is causing bacteremia to one where we start to get some of the meningitis effects, the swelling in the brain, etc. This is my really crappy rendition of the human body for today. Okay, so bear with me as we talk about this. All right. So what I want you to do as we're doing this, the important thing is not every single protein that I'm talking about. There is a couple that I do want you to know in particular, but I am giving you names to provide context here. Okay, so I'm going to refer to something as CBPA. You don't really need to know what CBPA is. You need to know more about what is it an example of, and what does it and other proteins like it actually do. Okay, so this is just to provide context more than anything else. So strep pneumoniae, the majority of incidences often occur through direct contact. We find that it can have multiple portals of entry. And a portal of entry is basically how does an organism gain access to the human body. Main ones, oral fecal route, cuts, scrapes, etc. In the case of strep pneumoniae, the most common forms, at least what lead to pneumonia and then meningitis, is going to be through nose and mouth. Okay. So we're going to consider this then, for the sake of today's conversation, this what looks maybe a bit wispy is mucus. It is not slime, it is mucus. Anybody know that movie? Damn it, guys. Princess and the Frog, guys. All right. Oh, yeah. Okay. This will be our mucus epithelium. And then below that, extracellular matrix, connective tissue, etc. Below that, we've got our bloodstream, or blood vessels. We'll consider this the endothelium. Also our blood-brain barrier. Okay, so beyond that would be then the brain. So when strep pneumoniae infects, it primarily comes in, and the first thing that it comes into contact with is mucus and mucous membranes. Now, any portal of entry, normal portal of entry, is in some ways covered with mucus, and that's a really good thing because the goal of mucus is to trap and to block microorganisms from being able to attach to host cells. We also find in mucus that there are high concentrations of IgA. Anybody recall what the Ig stands for, or what another term for that would be? I've drawn a Y-structured protein. What's that? Antibody. So IgA is one of those subclasses of antibody. They are most abundant within in mucus, and its main role is opsonization. What's opsonization? This is a term that we've talked about before. Opsonization. So I'll draw an example. Here's strep pneumoniae. Here's it being opsonized. What's opsonization? Not attachment.
When all the antibodies attach and prevent attachment.
They prevent attachment. The term that we've used is the antibodies coat the pathogen. Okay, and we'll come back to this because we have talked about an additional connection between opsonization and some other parts of the innate immune system as well. So we'll come back to this a little bit later today. But I erased everything. Oh crap. Well, it is what it is. So high concentrations of antibody within the mucous membrane. Strep pneumoniae, when it enters with a capsule. It can enter as diploid or maybe short chains, but it definitely has a thick capsule around the surface of it. It's also got a couple of specific proteins on the surface. One is this CBPA that I alluded to earlier. The function of CBPA is it helps to bind strep pneumoniae to receptors on the surface of these mucosal epithelial cells. And that receptor, in this case, is known as IgR, or immunoglobulin receptor. The role of immunoglobulin receptor is not really that important right now in terms of its typical role. But what happens here is that once strep pneumoniae grabs or attaches onto that epithelial cell, what we find is it actually gets endocytosed and it transmigrates or is able to be exocytosed onto the other side, into the extracellular matrix that's just below these epithelial cells. When it's in the extracellular matrix, there are a couple of other proteins that strep pneumoniae encodes. One is known as enolase, or ENO is the abbreviation, and the other one is known as PAV-A. PAV-A binds to fibronectin. Enolase binds to plasminogen. And these are both parts of, both types of proteins that we would find in the extracellular matrix. Strep pneumoniae also produces an enzyme known as hyaluronidase. And this is actually one that you saw in the mini lecture. What is hyaluronidase and what does it do? It can trigger an inflammation response. You're absolutely right. That's kind of a side effect of its function, Kevin?
Something to the cell, does it like go through that tube?
Yeah. Yeah, absolutely. Jaden?
It digests hyaluronic acid.
Yeah, it digests hyaluronic acid. In other words, what it's going to do is it's going to start to break apart some of the junctions that hold these cells together, also breaks apart some of the connective tissue. And in doing so, this allows for strep pneumoniae to basically penetrate past this extracellular matrix and gain entry into the bloodstream, causing septicemia or bacteremia. Okay. This is where we start to see some of the more severe effects of strep pneumoniae. So one of the things that we find is that strep pneumoniae will start to replicate, and at high concentrations in the bloodstream, some of these cells actually undergo what we call autolysis. What's lysis again? Burst. So strep pneumoniae, and we don't exactly know, I should say, I don't exactly understand what the benefit of this lysis is. There are definitely hypotheses to the potential benefit or why an organism would evolve to lyse itself. But what we find is this autolysis is driven by a protein known as LIT-A, and it leads to the secretion of a number of different proteins from inside strep pneumoniae, some which actually have function inside the bloodstream, and then some that just seem to trigger a very strong immune response. And that triggering of the immune response actually seems to be really important not only to the ability of strep pneumoniae to colonize inside the host, but it also contributes to its pathogenicity, or the disease that it ends up producing. One of the enzymes that is secreted is one known as pneumolysin. And pneumolysin is an exotoxin. And we'll come back to the importance of an exotoxin later on. Okay. In the name, it's a lysin. That means what, again? Lysis cells. In this case, this is what we would call a cytolysin, or sometimes the term that's used is a hemolysin. It is a protein produced by strep pneumoniae that ends up lysing red blood cells. And there is some benefit to that for the microorganism. We won't cover it right now. Maybe we will on Monday, but if anybody has further questions about it, we can address it later today or Monday. Okay? Just leave it at that for now. We do find then that some of these strep pneumoniae will eventually penetrate the blood-brain barrier if left untreated. It does so, again, taking the CBPA protein and binding it to a protein on the surface of these endothelial cells, and this protein is known as RPAF, or receptor for PAF. Again, don't worry about the function of it. But what we do know is that once CBPA binds to RPAF, the endothelial cell starts to undergo some structural change. Again, what we find is this interaction here triggers the host cell to endocytose strep pneumoniae. And again, strep pneumoniae will transmigrate from one side to the other, now making its way across that blood-brain barrier. Okay. We won't get really much more into depth in terms of the pathogenicity or the symptoms here right now. But one thing to point out is the brain is actually immunodeficient. It's kind of a protected area from the immune system, and you could probably see where this might actually be a benefit. As we've said, some of the parts of the innate immune response include, as Kevin was pointing out, inflammation. right? That is a common part of the immune response. And inflammation can be damaging. That's not something that we've talked about in great detail, but the secretion of cytokines at high concentrations can actually lead to tissue damage. It has some negative side effects, and you could, I think, start to see where damaging nerve tissue, brain tissue, etc., might start to be a troublesome phenotype. So in the brain then, what we can find is that strep pneumoniae, and being in this immunodeficient area, is able to thrive, able to replicate, and now can start to produce additional lysins, etc., that might lead to some of the tissue damage. Okay. So that's the general story. I want to first off see, are there any questions on this pathway here? Danica.
As of like what step would someone start to be treated so it wouldn't like keep going?
That's a great question. Probably at the point where you'd start to see some symptoms associated with it. And my guess is you probably wouldn't necessarily assume that it's strep pneumoniae or something like that until you get the bacteremia. Most of the time, though, we don't notice it until you start to get some of those signs and symptoms of meningitis, in which case you're already kind of behind the curve. Now, there are vaccines. There are vaccines to prevent this, and we'll talk about, they're known as conjugate vaccines, so we'll talk about that a little bit on Wednesday. But yeah, it's one of those diseases or illnesses that we usually don't know until somebody starts to present actually with the meningitis. Sid.
So what's the PAV-A and
The ENO? Yeah, so enolase and PAV-A are two other surface proteins or membrane proteins from strep pneumoniae that help it to attach to proteins within that extracellular matrix. So it helps with colonizing the ECM. Any other questions? Go ahead.
So are all these like proteins like the
Hyaluronidase?
yeah, does that come when like strep pneumoniae is present, or are they always there?
Yeah, that's a great question. These proteins, hyaluronidase, pneumolysine, ENO, PAV-A, LIT-A, CBP-A, those proteins are actually proteins that are encoded for by strep pneumoniae. So those are ones that you would only see in a strep pneumoniae infection. But the RPAF, the IgR, these are going to be present on these particular cells all the time.
Because can those like are those receptors for other?
Yeah, there's so, and that's the thing that we find a lot with illnesses or like a viral infection, for example. We oftentimes talk about there's a viral receptor on the host cell. Well, that receptor is just kind of by circumstance is a way for the virus to attach and to gain entry into the host cell. But these proteins do have functions for homeostasis, I guess. So the IgR, for example, its actual function is to take the immunoglobulin A that's being made or produced by B cells and to transfer it from basal epithelium and to actually secrete it across those epithelial cells so that it can get into the mucus. So the IgR is really just a transport protein for immunoglobulin A. That's its typical function. But the term that we sometimes use is in this case, the bacterium has usurped or has come up with another role for it that is not the one that the host has originally intended for. Does that make some sense? Other questions? Okay. So out of all of these proteins, the one that I really do want you to know about by name is this hyaluronidase. The rest, I think it's best to know them by category. We'll come back to that in a second, okay? But you don't need to know the pneumolysine. You do need to know things like what a cytolysine does or what a hemolysin does. May I erase this? Is that okay with everyone? Okay. So what I wanted to do, at the bottom of your page, you see a wonderful rendition of strep pneumoniae looking something like this. What I wanted to just do here is just to label for you a little bit of the structure so you know where do these proteins reside within that bacterial cell. So for example, on the surface here, what is this structure that helps to prevent phagocytosis? Yeah, it's a capsule. Great. And then below that, this area here would be our cell wall, a thick cell wall, as this is a gram-positive organism. We actually find that that autolytic or lit A protein localizes in the cell wall, which should make some sense given its function. It functions a lot like lysozyme and some of the bacteriophage lysins that we've talked about, and to degrading that cell wall and leading to the autolysis. That CBPA protein we talked about is a membrane-bound protein, as are the enolase and PAV-A. And then finally, some of these proteins are interior and then are secreted in some way. Hyaluronidase is one example of a secreted protein. I'm just going to abbreviate this for right now. That one actually gets secreted through protein channels. The pneumolysin, on the other hand, is a bit of a different story. It doesn't necessarily have a secretion system. It's released from these cells when lit A blows a hole in the cell wall and allows for that pneumolysin to be secreted. Okay. So those are kind of the major role players. There's one that's not on here that I'm going to bring up because this actually becomes one of the questions that I'm asking. And this is known as IgA protease. What is a protease? We've used this term a couple of times in the semester. ACE means it's an enzyme. What is a protease, though? If you hear prote, what does that make you think of? Protein. Protease is an enzyme that breaks down protein. Which in particular here? Which in particular is this one going to break down? IgA. So, think about the possible effects of that. If you think about the effects of IgA, or the role of antibodies in general, if you think about an organism being able to secrete such a protease, think about the benefits of that in helping it to avoid immune detection. Okay. All right. So here's what I want to do with the remainder of our time then. What I'd like us to do is to start to talk about more generally some of the classes or categories of enzymes that were, or proteins that were brought up in the mini lecture. For example, I said a few times that CBPA is involved in binding to host cells. What term from the mini lectures refers to proteins that are involved in binding to a host cell? Adhesins, exactly. So binding or attachment to a host cell, where we've got a receptor on a host cell, and then a bacteria in this case is binding to it to colonize, that is an adhesin. CBPA is not the only one though. What are other examples that we talked about here that are examples of adhesins? PAV-A would be one, yeah, absolutely. One more. Enolase would be the other one, yeah. So all three of those have a role in helping the organism to attach and allow it to colonize the host organism. Some people in their journal entries brought up that Strep pneumoniae does have a pilus or pili that will help with attachment, and that absolutely is true. We could define that as well as being such an adhesion. Okay. The other term was invasive. What is an invasive in general?
It allows an enzyme that allows organisms to pass certain barriers.
Good. So it allows organisms to pass certain barriers. And we actually see two examples of invasins in what we talked about. All right. So think about the different types of barriers that were passed and how Strep pneumoniae got past them. What's one example? The hyaluronidase would be one example. Absolutely, Abby. So that's one that's involved in breaking down that hyaluronic acid, allowing for the organism to get across that, impart that epithelial barrier, allowing it to get into the bloodstream. What's the second one? Pneumolysin? Not in this case. It's a good guess though.
RPAF?
That's the receptor, but what's it binding to on the bacterial cell, Kevin?
CBPA?
CBPA is the other one. So in fact, we can define, and this is a great case in point, some of these proteins are not just in one category. CBPA is involved in the adhesion, but what else did we say about it? Once it binds to RPAF, for example, it triggers endocytosis. It triggers the cell to basically bring it in. That is an invasion process, and it allows for not only the endocytosis, but the exocytosis out the other end. So that right there would be our second invasion process. Okay. Okay, so given that, we have essentially covered about 5 or 6 questions on here already. So